Hello, today we're going to discuss DNA replication. Okay, so what is DNA replication? It is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. DNA replication is made possible by enzymes. So enzymes are a substance produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. The suffix ase is used in biochemistry to form names of enzymes. The most common way to name enzymes is to add the suffix onto the end of the substrate. For example, an enzyme that breaks down peroxides may be called peroxidase. The enzyme that produces telomeres is called telomerase. So anytime you see that ase suffix, that helps you figure out it's an enzyme, it's a protein. Alright, so AP biology version of DNA replication. So first step of DNA replication is topoisomerase. Again, you can tell it's an enzyme because it ends in ase. That controls the rate at which the unwinding of the DNA happens ahead of the replication fork. It helps relieve the tension caused by the unwinding of the DNA. Next, DNA helicase unwinds the double helix of DNA, causing the replication fork to occur. DNA helicase breaks the hydrogen bonds that join purine and pyrimidine nitrogen bases. Third step in DNA replication. Single-stranded binding proteins stabilize the now separated parental DNA strands. Single-stranded binding proteins ensure that the separated parental strands do not reconnect. So that's super important because if they reconnect, replication will not be able to happen properly and you would get a bunch of problems. Okay. Fourth and fifth steps of DNA replication. So RNA primase adds a short segment of RNA or RNA primer to each of the separated DNA strands. So you can see that in the top GIF. And then the RNA primer then acts as an indicator or starting point for DNA polymerase to begin adding the appropriate new nucleotides that are complementary to the parent strand. And you can see that in the bottom GIF. Alright, direction of replication. DNA polymerase reads the parent strand going from the 3' to the 5' direction. DNA polymerase synthesizes the daughter strand going from the 5' to 3' direction. So it reads 3' to 5', but is making DNA 5' to 3'. The reason they're called 5' and 3' is just by you have to count your carbons of the nitrogen bases a certain way, and basically one end when you're counting around this carbon ring, there's 3' at the very end of one and 5' at the very end of another, so that's why they name those 3' and 5'. Looking at the replication fork on the right, you can see that DNA polymerase, no matter what, is synthesizing from 5' to 3'. But for the lagging strand, as it's called, it can't do it in one big piece. It has to do it in a bunch of little segments called Okazaki fragments. Then the polymerase shifts up, makes a fragment, shifts up, makes a fragment, and then that becomes one big piece. Okay, sixth and final step of DNA replication. DNA ligase seals the newly formed strand of DNA to its template strand. Okazaki fragments are sealed together. Hydrogen bonds are formed between nitrogen bases. And then in both of these pictures, you can kind of see how this whole process is just summarized at a more advanced level.
Thank you for watching. Please share and subscribe, and remember to spread fragments.